Yeah. Okay. All right. Um, okay. So good morning. Um, so I'm going to talk about resting state today, and then in some way it will be a very brief overview, again, which is like an hour and a half or so, and then um, again next week I'm going to cover each of these parts for like 2 hours, right? So um, I I may have mentioned to many of you that when I first started my PhD, um, because I was trained in engineering, I was trying to analyze the data. And so for resting state, I faced a lot of trouble. Okay, I still face some like trouble, but at that time, um, like every paper that I wrote would get rejected, and the comments were very personal, right? Like and mean, right? And so I always thank my advisor, who passed away a few years ago. So he always encouraged me, that he said, okay, you should not think of rejection as rejection, and the fact that people are rejecting your paper with such vicious comments suggests that they they cannot find fault with you, and it's like someone saying to you that you're ugly, right? There's nothing you can do. If someone said you cannot ride a bike, then maybe you can change and ride. But if someone said, then anyway, so I always thank him. Also, um, like when I was a student, generally across most places, you cannot become a faculty at that place unless you leave, right? So I was one of the very few exceptions, right? So that was like a big help for my career because it was very controversial, and yeah, I could have found a job perhaps in a company or so, but at a university would have been very difficult, right? But the fact that he actually asked me to write a grant when I was a postdoc, and the condition was that if the grant got funded, then I would become a faculty. So I became a so um, no pressure on any of you. So I finished my PhD in 3.5 years, and I was a postdoc for 10 months, and then I became a faculty. So there were several uh, students who started with me, who were students while I became a faculty, all because of him, right? So I always thank him for giving me such a encouragement and backing. And these are all my students who have like worked on resting state, right? And many collaborators. So far what we have covered with task activation is that you perform a task, like typically a block design. This is the ideal uh, reference. This is how the signal looks like. And then you can do a statistical test. And based on the statistical test, which is done on a voxel-wise measure, then you set a threshold using multiple correction and other um, analysis method. You say, okay, when you tap your fingers, these regions light up, and that is the sensory motor cortex, and therefore sensory motor cortex is associated with finger tapping. Okay. And like while this is correlation analysis, it can be done using GLM also, and these kind of things was done. I have also mentioned that the fMRI signal is like a convolution of neuronal activity with hemodynamics, and then we get the fMRI signal, right? So you perform some task, there is increase in neuronal activity, it's coupled with hemodynamics, and then you get the fMRI signal. So engineering, it is called the transfer function, right? So like you have input, you have output, you want to understand, you know, um, like this one, right? This happens to be our brain, and like one approach or many approaches assume that this is a black box, right? And then we would like to find the relationship between input and output. Okay, so input is the cause or the signal that goes into the system, right? So tapping your finger is the input. It goes through some black box, right? And then there is some rules that we don't know that we want to find out. And output is the effect, right? And so I would say that in most of most research, the idea is not just to find the relationship between input, but to find out what is this transfer function. And and the reason it's important because, you know, I could tap my finger this way and then you could see a response. I could tap in a different way and I would see a different response. That is never enough. What we want to know is that what is the relationship between input and output such that I could make a guess, right? You know, if instead of tapping my fingers, like if the stimulus was 20 seconds on, 20 seconds off, 20 seconds, if instead of that it was 40 seconds off, 20-second on, 20-second off, 20-second, what would the output be? Any guesses? What would happen, right? So in right, so in one case it is like this, and the response that I get is right. So this is input, this is output, right? And I tried this several times and it works. And then I decided to change something, right? I did, okay. Can you guess what the response would be? Yeah, Sanchit. This is input, this is, it's a simple question. It's, don't think I'm asking something difficult, right? So in this case, the response will be something like this, right? It's something, the assumption with this is that once you have the stimulus, the signal will go up. Until then, it's going to be baseline, right? And so that is trying to understand the transfer function, right? Can anyone think of um, other examples? Hmm? You can do that, but think of something not related to brain imaging. So I'll give you an example. So today morning, I went by a like a three-wheeler to Sangam. Sangam. Uh, if I had gone by a car versus, you know, a three-wheeler, what is the difference? Like the the experience is more bumpy, right? Because the transmission system of a auto is much better, right? So if a vehicle has good transmission, then it absorbs most of the shock, right? So that's the transfer function, right? So think of some other examples. So the transfer function, maybe I should say, is like a simple rule that describes what, you know, the object that you're studying does, right? In this case, what does the brain do? So, you know, it is maybe some way to describe how it will convert the input into the output, right? And if I put this in, right, what will I get out, right? So if I was to drink coffee every morning, then, you know, it would make me alert. But if I drank 5 cups, maybe the response would be something else, right? May not be that pleasant, right? So in some way, the transfer function will tell you if the response will be amplified, or it could also attenuate, right? Or it could delay, or it could filter, right? Like you have this um, like if instead of this classroom it was in a large auditorium, right, then the sound that you would hear would be perhaps different than here, right? So maybe anyone, so many of you have said that your background is in psychology. Can you think of an example in psychology? So maybe I will give you one example and then you think of another example. Um, Maybe, okay. So this is like phobias and exposure to, again, I don't really know how true it is. I'm just, so your input is a specific stimulus that is seeing a spider, and the output is that you have like a almost like a panic attack, right? So in that case, somehow the brain's fear circuit is getting triggered, right? And if you are a psychologist, perhaps what you would like to do is change the transfer function, right? Such that when someone sees the spider or a cockroach or whatever, you don't get that response. Yeah. So so that would be the example for understanding transfer function. But maybe maybe I'm making it too complicated. So maybe another example would be that um, you got, like, on Friday you got at 5 an email from your professor that we need to meet Monday morning at 9, right? Depending on your personality, or and your, like, what you think of your professor. Some may say that, maybe when I come Monday, she or he will tell me how amazing I am, right? Um, or it could be that, oh my God, right? My paper must be really bad, and I'm going to get a showdown how I have to improve, right? And then based on, condition one, or the first versus second, your weekend may be, completely ruined, or you may say that I'm so good, I always knew it, and now my professor is going to tell me on Monday, right? So in that case, right, if you become too anxious or worried, right? Again, I'm just giving an example. It has nothing to do, right? So then the idea is, if someone is getting very anxious for even little things, what can one do, right, to calm them down or make them better? But that, so again, this is not transfer function. This is changing the transfer function. But what I'm interested is understanding the transfer function, right? So if in this case you have an input and an output, is this the best input to use to study the output, right? Or study the transfer function. So what is the best input that one can design to identify the transfer function? So in engineering, we we call this like the transfer function H, because it determines how the input is related to output and vice versa, right? So if you know the transfer function, if you know the input, you can predict the output. If you know the output, you can predict what the input is, right? So then the question is, if we go back to this example, right? And instead of this, if my stimulus was, right, something like this, would you be able to predict what the response would be? Remember, like based on the stimulus duration I mentioned yesterday? Right. So the challenge with block design is that it is good for, you know, similar kind of stimulus, but it's not very good when the stimulus duration is very long, or the stimulus duration is especially when the stimulus duration is very short, right? So that is like, so with this, we are not able to capture the transfer function adequately, right? So then what is the best way to capture the transfer function? I had mentioned it yesterday. So if I want to find out what the transfer function is, what is the input that I should use to better understand, or which would capture my transfer function? Uh, like in what order? Like random order, periodic order, what kind of order? R- Right. So, but as I mentioned, right, for block design, because it's periodic, it's sensitive first to that frequency. So while you should be able to predict, make the linearity assumption and be able to predict for other frequencies, generally for shorter ones, the prediction is not good. So so the best way to find the transfer function is to give a random signal, right? And as I said, because with random signal, it has short duration, long duration, specific, like many different frequencies and so on. So that will give us a very good response, right? So another way to think is that um, if these brain signals are intrinsically, there is some kind of architecture. So even if noisy signal comes in, the response, although will be noisy, will have unique characteristics, right? Because they have unique transfer functions. So that is the basic idea with resting state. So this is like a resting state signal from the sensory motor cortex. And this, so you can see like there is the signal which is present. Again, this is just during rest. This is the respiration signal. But if you see, there are these fluctuations which are present, right? And as I had mentioned, on the first day, what we did was we used correlation analysis just exactly the way we did for task activation. We took this time series and correlated with every voxel in the brain, right? And what we got was this, right? So if you had to guess, can you guess which one is task and which one is rest? This is task, rest. How many say this is rest? Raise your hand. Okay, and this one is task. Why do you say this is task? Okay, good. Yeah, so, okay, good. So this is task and this is rest, right? So the main findings of this study was that the temporal noise characteristics in the brain were significantly higher, um, like in the brain were significantly higher than outside the brain, right? The outside the brain, like this is the noise characteristics, this is inside the brain. So significantly higher. Different brain regions like gray matter, white matter, CSF had different noise characteristics. So this one is white matter, this one is V1, the gray matter. You can see heart rate, harmonics of the heart rate, respiration rate, and then there is this very low frequency signal that was observed. And that these significant low frequency signal was present in the brain and was different between gray and white matter, right? This is gray, this is white. And that the left and right sensory motor cortex had significant correlation during rest, right? Between left and right. So there was significant correlation, and we could identify these sensory motor cortex just like we would do for finger tapping, right? And you know, we had also observed in the language and visual region, but there was a paper I had written, I gave up after like 6 tries because I simply could not get it published. So because Carl Friston, who perhaps is the most famous neuroscientist or neuroimager, had defined functional connectivity as correlation between different brain regions, and we were observing this during rest, we called it resting state functional connectivity, right? Any questions? Okay. So initially, not like there was a lot of criticism, and these are the early papers, right? So this is by Mark Lowe, this paper where he showed that you could see sensory motor correlation across multiple slices, right? So again, when I did the study, you could only scan one slice, right? So you could only scan one slice, and that is why it was very difficult. So we had to choose one slice from the sensory motor cortex, and you could only collect 512 up to 512 images. So instead of collecting 2 slices, I decided that I should just collect one slice and collect 512 images and then analyze it, because one would have more data points, right? And so what Mark Lowe did was they collected 2 slices, right? And then they showed that you could see this correlation across multiple slices. See, that is where when you work on controversial topic or a new field, like every little tweak that you do, you can publish it, right? So then, like Peter Fox, maybe some of you may know, like he was the founding editor of Human Brain Mapping. So he wrote this paper where he said that this is task activation, M1, SMA, um, because it's the ROIs are so specific, and this is for resting state, right? And he said something which I never had the courage to say, and that is that task activation is just a subset of resting state, right? So, like, and his point was in resting state, you could identify the primary motor, the supplementary. I'm so sorry. Okay. Okay. So, so, yes. Oh my God. Okay. One second. Okay. That's okay. Um, so he suggested that um, resting is a superset on which the task activation is based on, right? Then Dietmar Cordes and Beth Myron, they showed that there is a frequency structure to these correlation, what we had seen with the low frequency signal, right? And then Michelle Hampson showed that for the reading network, you could identify the reading network from the resting state network. Um, uh, I don't, okay, I thought it was published in Journal of Neuroscience, but maybe Human Brain Mapping. So these were like the first few papers that was published. Oh my God. I have no idea what to do. Okay. I think he called me so many times that my computer froze. Okay. So these were the early papers. So um, so here we showed that we could look at the signal and then reduce the physiological fluctuations by using digital filters. Um, we also did a study in animal models where we could show that there was synchronous oscillation in the cerebrocortical capillary after nitric oxide synthase, right? Again, these fluctuations could be modulated using neurochemistry. Then we did another study where we used hypercapnia stimulus to show that these fluctuations could be modified or modulated by using like subject breathing CO2, where there is a significant increase in blood flow. We also showed that in Tourette syndrome, like, does anyone know what Tourette syndrome is? Yeah, maybe so. Yeah. Can you speak a little louder? Right. So it can be, there are many different kinds of Tourette's. Like one is like motor tics, and the other one is language tics, right? Um, and then we also showed how these fluctuations um, can be modulated. But in around 2003, like about 8 years after, there was a paper published by Michael Grecius and Vinod Menon. Um, I think like um, you had a speaker a few years ago, Dr. Chandan Vaidya. So maybe she had worked with him, or they had worked together. So in that, they showed that these functional connectivity during rest and the default mode, they were related, right? This paper by Michael Fox and Marcus Rakel showed that the brain is intrinsically organized into dynamic anticorrelated functional network, right? And he's a member of the National Academy of Science. And so this was published in the Proceedings of the National Academy of Science. And then there were other paper like Vince Calhoun had um, one of the papers about ICA in resting state data and how it could be used, not in resting state, in task activation, and how it could be used. Vesa Kebenimi also used independent component analysis for fMRI. And then Chris Beckman and Steve Smith also used this in resting state data, right? So this is Christian Beckman, and that's Vince Calhoun. And this is just showing now that we could collect whole brain data. What they showed is that from one resting state scan, you could identify many different runs or many different network from a single run, right? Before, like for task activation, if I wanted to um, visualize um, where the sensory motor cortex is, I would do a finger tapping paradigm. If I wanted to see where the visual cortex is, then I would give a visual stimulus. If I wanted to do working memory, then a corresponding task. So in task activation, typically you need at least one run to stimulate that task, right? But with resting state, from one run, you can identify many different networks. So what is ICA, right? So based on what we have covered so far, right, for task activation, we make this assumption that, I have a stimulus and I have an analysis method, and that is how I'm going to get my results. But it should not be that way, right? The way it should be is that it should be determined by a hypothesis about the underlying cognitive process, right? And as I've tried to mention several times, like while listening to music or listening to auditory task or visual task or motor task, you can do like a block design or a simple GLM analysis. But for other tasks, like if you were to get a stimulus where like getting injected with different kinds of um, you know pharmacokinetic drug like cocaine and so on, the response would be very different, and maybe it would be dependent on the subject. If the stimulus was, let's say, a pain stimulus, right, it may also, right, the response won't be linear and maybe dependent on that. So while with GLM, one of the big assumptions you make with GLM is that you assume that you know what you will get for your response, right? That that may be true in many cases, but one can also argue that, you have already, like, if I did finger tapping and I chose correlation, I have already made assumptions about what my results will be, right? Because it will it can only be a scaled version of the input, right? And if there was some kind of other effect, then you will not see it, right? So this other kind of analysis method that has been developed um, is called data-driven method, right? So um, like with the data-driven method, you use the data to derive what the response is, right? So maybe another way you can think of it is that, like when we use GLM and correlation or t-test, what we are asking is, when you do the task, where in the brain is that response, right? But if I was to ask you the question, what does the response look like? Will you be able to tell me? How would you tell? Hmm? So if if one did finger tapping task and we did GLM or correlation, we can identify the sensory motor cortex, right? So that tells us where in the brain the sensory motor cortex is, or where in the brain is the region corresponding to finger tapping is. But instead of where, if my question was, what is the response? How does that response look like, right? How would you go about finding it out, Rahul? Okay, so let's say you found out where that response is, right? Can you now tell me what the response is? What what happened? You say, "Mm, oh, no, no, that would be good. So that's good. So how would you do that then? So you would find where the the region is active and extract the time series from those regions, right? And it will tell you like an approximate response, how it would be, right? And as you get better, you could um, average across region and look at the time series. Yeah, is that clear to everyone? No. Um, okay, maybe I can go back. Okay. Um, so let's say this one. So we did finger tapping and we found this region, right? And so this is where in the brain this response is. Now I wanted to know, how does, when someone tapped their fingers, how did the signal look like? So what I can do is, that's what I was trying to tell you yesterday, right? If I gave you the coordinates, the x, y, and z coordinate of this 3D plus time data, then you should be able to plot the response, right? And that would give us an approximation of what the response is. Is that clear? Yeah, no? Yeah, okay. So So while one can do it, the other approach which has been used is like, it's called independent component analysis, which is like principal component analysis. Has anyone heard about, does anyone know what principal component analysis is? Yeah. Uh, can you simplify it a little bit for such that others can, maybe Rahul? Right, right. Okay. How about you? Yeah, yeah. Right. Okay. 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 Um, so in principal components, as they all mentioned, like you take a set of, when you have a large number of variable, then you can decompose into a set of basis function where each one is independent of the other, right? And when they, sorry, each one is orthogonal of other, right? And when like two components are orthogonal, then the correlation between them is zero, right? But independent component is that orthogonal doesn't mean they're independent. So independent component analysis is another um, way to analyze the data. So in this case, what independent component analysis does is it will take all the data, all the time series data in your brain, and then try to find out a set of independent component, right? So, um, right. So this is something like this, right? So you may have a source like this, another this, right? A set of basis functions, and then they are getting mixed up, and then you have to identify these ones, right? It's the other analogy which is used about ICA is, have any of you heard of the cocktail party problem? Yeah, say what it is. Very good. Yeah. So, um, so what we have to then do is, right, um, we have to, we basically identify unique time series, right? Because like if when someone is telling your name, it doesn't have any unique temporal information or spatial information, but the fact that someone is mentioning your name, you can like very quickly like uh, um, identify, and you can also, if you want, right, you may be talking to someone, but you can eavesdrop on someone else. Maybe it's difficult, but someone can do, right? So in this case, what we are trying to do is try to find in the brain what are the signal sources, what are the dominant signal sources, okay? And like using ICA, this is this is for task activation. Um, this is the spatial map, and then you can look at the components and then do the frequency spectrum and then find that, okay, this is relevant to the task, and then find these ones, right? So that's the visual cortex, that is the heartbeat, breathing signal, head motion, vascular oscillation, and things like that. So that was done for task activation. And then this is Melodic's FSL's version of ICA. You can take, this, and this is your time course. Then you can look at the frequency spectrum, right? And then you can say, okay, for this one, this is the task response, right? This is perhaps some fluctuations. This is definitely physiological fluctuation. Anyone wants to guess what this one is based on what I mentioned yesterday? Head motion, right? But some of the limitation of ICA is that it is like very popular to use, but it cannot test hypothesis, or it's at least not directly. And you know, the most dominant component may not be very, like, it doesn't mean that the most dominant component is something significant. It could be the most dominant component typically can be head motion, right? So you have to go through the data to find interesting components, right? So while like this is one way to analyze your data, there are other ways. This is called a brain parcellation method, which has been developed by Thomas Yeo. It's called the Yeo Atlas, right? And what he did was he took the entire time series like you do in ICA, and then they used clustering method, right? And they used hierarchical clustering using a thousand subject. They identified these 7 networks, right? Visual, somatomotor, dorsal attention, ventral attention, limbic, frontoparietal, and default mode, right? So then one can use this to identify these network, or you can, since these have been derived from 1,000 subject, you can take the atlas and test it on your data set, or use this region as region of interest for your data set. There is other method called gradient method, and again in this case we use gradient, and then we can also identify these gradients and find out different components corresponding to different regions. So some, like, maybe Monday or Tuesday, I'm just going to cover about all this analysis method one by one in more detail. So one of the question is that, what gives rise to these physiological fluctuations where we get these, connections. So one of the first studies that we did was in animal literature. people had also observed these fluctuations, but in animal, they would only have a single physiological signal. And what they had observed was that if you give a rat 5% CO2 or 2% CO2, the blood flow increases by 50 to 70%, right? And these low frequency physiological fluctuations that they had observed would be significantly diminished. So we thought like we could give 2% or 5% CO2 to humans and then see what would happen, right? In humans also, when you breathe 5% CO2, your blood flow will increase by 50 to 70%, right? So this is finger tapping experiment. This is pre-hypercapnia, like when someone is breathing room air. This is when someone is breathing 5% CO2, like, so they breathe 5% CO2 for like 9 minutes. And after the signal stabilized, we analyzed the last 5 minutes, right? So that the signal was at a steady state. And here we could not see these fluctuations. Also, the low frequency fluctuations was diminished. After breathing CO2, they were like, they rested for like 5 minutes, breathing room air. And then we scanned again and we could again identify these fluctuations, suggesting that it's not an artifact, but perhaps it has something to do with, like, there's a biophysical mechanism which leads to these functional connectivity, right? And this is Vesake Venemi. He was my first postdoc. So he was anesthesiologist, or he is an anesthesiologist. And so what he was doing was that they were giving children thiopental anesthetics, and they observed large fluctuations in the visual cortex. Again, this is like 25 years ago. So at that time, you could also only scan one or two slices, right? And so when they were studying the visual cortex, they saw these fluctuations go up. And yeah, so to help understand and analyze the data, he had come to Wisconsin. And so we saw that these temporal correlation was higher. Also, when you made a FFT, the magnitude was much higher, right? And this is a study by Scott Peltier and Xiaoping Hu, where what they did was they took human subjects and then they gave them anesthesia, like sevoflurane, which is commonly used now. So first was awake. So during awake, you could observe left and right connectivity and connectivity between left and right. With light anesthesia, you could observe connections in the left side. You could observe connection on the right side, but you did not observe connection between left and right, right? So this is connection between, here both left and right is connected. Here only one side is connected, but not the other side, right? If they had placed seed in this region would have been connected, but not this side, right? So it was on one side. With when the anesthesia was increased, right, then connection both within and between was significantly affected, right? Again, suggesting that they have some physiological meaning, right? Also, because different anesthesia work in different ways, in some cases, there is an increased connectivity, and in some cases, there is a reduction in connectivity. Okay. Then this is paper by Justin Vincent and Marcus Rakel, um, and this was published in the journal Nature, where this is in monkeys. They collected fMRI signal as well as electrophysiological signal, right? So what they showed was that you could observe a connectivity pattern during light anesthesia, and you could see the electrophysiological signal, but when the anesthesia depth was increased, the connectivity pattern here was significantly reduced, right? There's no bilateral connectivity. Also, the EG signal pattern was different than in when the anesthesia was, the depth of anesthesia um, was low. And again, these results indicate that coherent system fluctuations probably reflect an evolutionary conserved aspect of brain function organization that transcends levels of consciousness. So again, I want to emphasize that task activation has many advantages, right? But you can all see that in many cases, you cannot do task activation, right? If I asked okay, maybe a 5-year-old would do, but if you ask like a 2-year-old, right? Or if you ask someone who was in a car accident, right? Or had a stroke or was in coma, right? Um, and for many neurological disorders or even for psychiatric disorders, right? If you ask a schizophrenia subject to do something, it may happen that they will do something that you didn't ask them, right? So it has many limitations, right? Like in many subject, it cannot be performed. So this is showing in infants by Hanzang, where they showed that using seed-based, you can identify the motor cortex. You can also use ICA to identify motor cortex, right? Similarly, the visual cortex, seed-based, this is ICA. This is for default mode, um, right? This is seed-based, this is ICA. Again, if one was to design a task, it would be very difficult to do such a task, right? This is another famous study by um, Damien Fair, who won actually the MacArthur Award like 2 years ago. So this is showing a connectivity pattern in adults and children. And then this is showing differences between adults and children, and then using a network kind of analysis, showing how the different regions are connected. And in adults, how many of these connections which are not formed in children has been, um, you know, has been formed in adults, right? So this is like showing how with development, these network properties are changing. This is a study by Xiongong, and in this they showed connectivity pattern in major depressive disorder. And in this is an interesting study because uh, till that time, most of the study for connectivity would either show an increase in connectivity or a decrease in connectivity for different groups. This study showed that there are some regions where the connectivity increases, and there are some regions, like in blue, where the connectivity is reduced, right? In MDD patients compared to healthy control. And they also used a graph theoretical approach to display like the thickness is increase in connectivity. This is in reduction in connectivity. This is another paper by Helen Zuo and Bill Seeley, right? And this is a good paper because many studies had showed, right, between like Alzheimer's and healthy control, there is a difference. Between schizophrenia and healthy control, there is a difference, and so on. So what they did was they wanted to ask the question, if we take 5 different neurodegenerative disorders or subjects with neurodegenerative disorder, will there be differences between them or will they be similar, right? So AD, many of you know what AD stands for? Very good. Anyone knows what FTD stands for? Oh, pretty good. How about SD? Okay, I don't know either. The what? Okay, and so these are 5 different neurodegenerative disorders, and in this case they wanted to see if there was something specific to each of these disorders, right? So was there like, could we differentiate AD from frontotemporal dementia, AD from semantic dementia, and so on? And what they showed was that each of them had unique patterns, right? Unique connectivity pattern, which could be used to differentiate between them, right? So far, okay. Um, so let me think something. Can you think of a task, or can you think of a population where, um, like, you would do you would do resting state but not task? Good. Any Very good. So like coma, right? Coma. Anyone else? Okay. Right, right, right. So good, good, good. Anyone else? Mm-hmm. Right, right, right. Okay. Mm, a- okay. Anyone else or anything else? Any other disorders, Shreyans? We covered infant, right? Uh, I would argue that almost any disorder that you can think of, you can do resting state, right? You can also do task activation, but because like the additional if you're going to do task activation, the additional cost is maybe 10 minutes, right? So maybe you could still do. Okay, let me continue. So the next thing I think I had briefly covered. So there is some debate as to what is task activation map and what is resting state connectivity. Some have suggested that the resting state map is a superset of task activation. That's Peter Fox. Others have suggested while task activation is used for functional localization, um, resting state, like functional connectivity, is used for functional integration. Right? And this is the paper by um, Jin-Hyu Xiong and Peter Fox. And this is task activation, and this is resting state. And because they showed that there is so much overlap, um, they suggested that this is a um, superset, right? So Peter Fox also has developed something called the brain map, right? So brain map is a database of, I don't know how up to date it is, but it's a database of all the neuroimaging studies, right? I should say all neuroimaging task activation study. So what they do is that, if there is finger tapping, right? They will say, okay, when this region is activated, what other region is also activated, right? So they call it co-activation, right? So by doing that, what they have done is they have developed this map where if you type a particular term, you will get what other regions have been associated with that region. Okay, is that clear? Okay, so what they did next was with Steve Smith, they took every information and then they did a independent component analysis, right? So you have A is associated with B, A is associated with C, then you can go and see that B is associated with other, right? And then they did a ICA, and what they found was that they could identify these network, right? They identified this network, they identified this network, right? Defaultment, and so on. And so that is on the right side. On the left side is a set of not many, maybe 50 subjects, their resting state maps. Okay. And so what they found was these two were so similar, right? And so by using coactivation, they again showed that this connectivity pattern is dependent on the resting states architecture, right? So what we did was, again, so we can make a coactivation matrix by using which one is connected to which region. So this is for 140 region. We got the coactivation using neurosynth. This is looking at correlation, and then we are plotting this versus this, and the correlation was 0.72. So the correlation was very high. So what we wanted to do is that, okay, like Steve Smith and others have found that there are, significant overlap between the two. What we wanted to see is that if there was some difference between coactivation network and resting state network, right? And by using graph theory, this is what we observed. This is like modularity using graph theoretical network. This is the correlation network. Many of them are connected to others. This is the coactivation network. And this is if it was a random network, right? And basically what we find is that during task, the coactivation has higher global efficiency, lower mean clustering coefficient, lower modularity, high degree in the thalamus, and during rest, there is lower global efficiency, higher mean clustering coefficient, higher modularity, and high degree in the left temporal lobe, right? So these are like some way to interpret the data. The next The next topic I want to cover is like calibration, right? And that is, as I have mentioned several times, the fMRI signal is dependent not just on the neuronal activity, but also on the underlying hemodynamic response function. And if you get high signal changes corresponding to a task, people generally assume that it is due to neuronal activity, but that is not necessarily the case. Typically, it may be due to the underlying hemodynamics, right? So if you have a large vessel, then the signal would be large, not because there is more neuronal activity, but because of the underlying hemodynamics. And the way people have done is um, they have tried to scale this response, right? So what they have done is they have done like a breath hold paradigm where you ask someone to hold their breath for like 15 to 20 seconds, or make them breathe carbon dioxide for 15 to 20 seconds. And when you hold your breath or breathe CO2, what happens is there is an increase in blood flow, and only blood flow, not neuronal firing, only blood flow. So for every voxel, you can tell which voxel will have large flow, which voxel will have moderate flow, and which voxel will have less flow, right? So you can identify this on a voxel by voxel basis, right? And so you can find the change corresponding to task, but you could also find the change corresponding to breath hold, right? And then this is how you could scale it. Okay. Um, so this is similar to standard deviation of task divided by standard deviation of breath hold. Okay. And so our hypothesis was that we could do this, but what would be simpler is that the resting state data, those subject did not perform any task. And so if we simply divide the standard deviation from rest with the standard deviation during task, maybe that is another way to do the scaling, right? So I used to always joke that even if you don't believe in resting state, you should collect one resting state scan because it will help you clean up your task data. Not many people find it funny, but yeah. So I think as Sanchita was saying that like with aging, there are issues, right? And there can be changes in the underlying hemodynamics, but there is also changes in the gray matter volume and the response and so on. So what we did was we did a motor task, right? And this is a DSST task, which is a cognitive paradigm digit symbol subtraction task. And then we made the subject hold their breath, and we calibrated for gray matter volume and breath hold response, right? Some of the regions which was like had large changes, um, like after calibration, those responses went down. And then by using ALFF and gray matter volume calibration, we can get very similar responses, right, for both of this. Again, the point is sometime you can use this signal like from rest um, to understand the fluctuations and use it to clean up your task data, right? If the task data, if the task data is very noisy, then it is worth checking what is happening to the exact same voxel during rest, right? If even during rest it's also noisy, then maybe you should be a little more careful in how you represent your task data. And again, with resting state, you don't have to do a breath hold or give a hypercapnia stimulus, right? Again, hypercapnia stimulus is a direct stimulus, but in many cases, um, those of you who were subject, like being in the MRI is not the most comfortable place to be in. And some people are claustrophobic. And on top of that, when you have someone put a mask, it becomes difficult, right? So that is one challenge. And for many people, especially old people, they may not be able to hold their breath, right? And even in clinical populations. So again, the point I want to make is that um, resting state data has other application, and even if one's primary focus is not in functional connectivity, we can use the resting state data to clean up your task signal, right? You can like look at the frequency properties and then um, analyze accordingly. So um, as I have mentioned before that um, like many of my or most of my papers would get rejected and I would have to try many times before it got published, um, and also with grants and so on. So it's one thing to have your papers rejected and your grants rejected, but it's a whole different problem when the editor-in-chief of the main journal of your field, you know, write a paper, does the brain have a baseline why we should be resisting rest, right? When the editor-in-chief is trashing your work, um, he is no longer the editor. So I think what happened was Paul Fletcher, who was the editor-in-chief of Neuroimage, um, was very uncomfortable, was not happy that so many people were submitting papers on resting state, right? So he decided to write this paper. And then actually it's a good paper. Like if anyone is interested in resting state, you should like read some of my papers, but read this paper also, right? So the main the question was, what does resting state mean, right? And their point was everything we know about neuroscience has been from task activation, right? And so why bother with something, you know, um, like with resting state? Like since task activation has been so good to neuroscience, why not just stay with task activation? The second point was, um, how do we know what the ground truth is? How do we know it's not an artifact, right? Um, and because, and I think like he has a point in a sense that if we don't know what the signal is, then if there is an artifact, it's not that easy to remove the artifact, right? Um, the but the other thing that they said was that, oh, all these studies are done on 20 to 30 subjects. At that time, even task was done on 20 to 30 subjects. So the editor said that if a study was done with maybe 500 or 1,000 subjects, maybe they would start believing, right? So, um, you know, I don't know, how many of you have published some papers? Anyone? All right, okay. Good luck to all of you. Um, so those who have published paper know that it's not easy to argue with reviewers. You know, if you argue, you know, uh, well, first of all, when a reviewer is reading the paper, you know, no matter how good a paper is, if they don't understand it, they will never say, oh, Sanchita wrote such a good paper that maybe I don't know something and therefore I should accept the paper. It will always be, oh, I don't know what this person is talking about. So you reject, right? And you cannot like if you say that, don't you know this, then it would lead to automatic rejection. So, so because you cannot argue, so what I did was, I was also, I should say, a little worried that would it hold true for multiple sites, right? Although I had like data, as I said, from different people who would send me or ask me help for data analysis, and some would come for short stay. Um, so what I did then was I asked every group who had published on resting state till then to share their data. And asking, for data is like, at that time, maybe even more controversial than resting state. But luckily, we had several groups who shared the data. Like I would I would say that most said no, but there were a couple of researchers I should point out, like Zhang Yufeng from Beijing, Normal, and many of you may have heard of Randy Buckner from Harvard. So they gave 200 subjects each, right? So, and then I had like 700 or so. And then I asked the same people again. I said, I'm going to publish a paper with 700. You can join, and even with 30 subjects, you would have a good paper, right? So then I had more than 1,000 subjects. So I will always be grateful to these people because, again, so we have 1,000, more than 1,000 subjects. Again, these are all publicly available. There was one thing I did which I never thought, or at that time I didn't think much of it. So what I did was, like, so people would come and ask that what would happen if the filter was this or filter was that. So I thought like, okay, I should also put the code along with the data. So if someone wants to run the code, they could basically reproduce the figures. And I think that became quite helpful, right? Many people could just use the code, run their data. So, so I'm forgetting the main thing. So this is data from like 13 or 14 different countries across, like from North America, Europe, Asia, Australia. And the data was collected using Siemens, Philips, GE, and Varian, right? And then 1.5, 3 Tesla, 4 Tesla, and 7 Tesla. So, like, different manufacturers, different field strength, different ethnicity. The scanning was done at different times of the day. Even the pulse sequences were different, although most was EPI. There were different sequences that was used, and we processed them in exactly the same way, all the data, right? And then when we used ICA, we identified these 20 components, right? Again, this is from 1,000 subjects. This paper only got rejected 3 times. So again, because we had so many data, we could ask very specific question, right? We could ask, is there differences in male versus female, right? For different brain region, is the connectivity pattern different? We could also ask the question, like, is there differences in connectivity with age, right? And so, again, by using this, we could look at all these changes, right? And since then, there has been many other effort where people have used much more data, right? This data by Vince Calhoun, they used 47,000 subjects, right? And the idea, I think they are going for is that eventually they want to, once you have such a large data set, you want to integrate with maybe the genome information and find out important, like which region is association, right? Like GWAS that they say. No. Um, right. So how is it possible that the difference in scanning parameters did not have? So why actually, when we did, the biggest difference we saw was, make a guess, what for? So the biggest difference we saw was an aging subject. Like someone had sent us like old subject, like 80 year, like between 60 to 80 years of age, and in those the connectivity pattern was significantly reduced, and they had also used a 1.5 Tesla, which made it even weaker, right? um, I did not want to exclude, because it would have been like, what would I tell the person that, hey, you're doing aging research on 1.5, and it, that's not a good enough reason, right? So the age was the biggest factor. Then was the scanning, the field strength. In terms of imaging parameters, like if you are using EPI, unless let's say you are using 3 Tesla, do you know your imaging parameters? So TR is 2 seconds. How much is this after TR? The most important parameter is echo time, right? How much is the echo time? How much? 130, right? So it's like if you had used 60, then maybe you would have a, like, the signal would be very low and very noisy, right? But most try to use like the like a parameter which is reasonable, right? So that is why I don't think it had a difference. You sound skeptical. I would say please try the data. It's publicly still available, you know. So there again, as I'm saying that, so there is this study where they have used 47,000. There is one using the UK Biobank, which has now more than 100,000. Um, and then there is another, this is like on depression, which has also more than 1,000 subject. Again, here what they're doing is they're using the depressed data from more than 1,000 subject, and what they are saying is that by analyzing the resting state data, they are saying that there are two subtypes, right? And the two subtypes have these different characteristics, right? And they also speculate how behaviorally or clinically they have differences. Any questions? Any? Right. So we typically have a script, like we have like a 2 to 3 sentence that we read to every subject, right? It would be like, you know, try to lay still, try not to move your head, just like relax. Um, what else? Maybe um, either keep your eyes open. Um, but again, like I don't think there's a large difference between eyes open versus eyes closed. I personally ask the subject to keep their eyes open simply because I have been a subject many times, and I have noticed that if I keep my eyes closed, I tend to fall asleep. So that is the reason. But I don't think there's a big difference. Um, what else? I think that is uh, all we give. Right, right. Right. Uh, yeah, yeah, yeah. No, no, I agree, right? But but that is what I tried to mention that the fact that we are still getting so similar uh, connections, right? It's very unlikely, right? If you um, got scanned today and then I got scanned, it's very unlikely that we would be thinking about the same thing, right? And yet we the connectivity pattern, you know, many regions mine may be a little weaker than yours, but the connections hopefully will still be there, right? So um, then what does it mean, right? And why do we see it? And does anyone have any guess? Why do we see this connected pattern, even though we all agree that what Rahul would think in the scanner is perhaps very different from what Shivani would think, and so on, right? So then why do we see the same connection pattern? Right, right, but even daydreaming has a temporal sequence, right? Like, so let's say um, you were scanned and you were thinking of, let's say, how good the lunch was. But it's very unlikely that someone else who was scanned would think that, unless the lunch was really, really good. Right. So, does anyone, again, I really would like all of you to participate, right? Why do you think it is happening? And anyone else? Uh, uh, Right. Okay, good. Good. Right. 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 And anyone else? Any other thought? Say something. Yeah. Why do you think it is happening? I don't. So, I wish I knew the answer. I have like everyone has some guesses, and my guess is as good as yours. So you might as well tell. Right. Um, so they were, yeah. So I think what she said. Okay, she's saying that the fact that you're being put in a scanner and in a very confined space, and the process is the same, and therefore the input would be the same. Um, that that may that may be, but I would say that perhaps only it would have a transient effect, right? Only for the first couple of minutes, even if it is that long. Um, so um, I don't really know why, but what I can, like, my guess is that um, the input in this case doesn't, like, for all of us, it's a random noise. And what we are measuring is the transfer function, right? And because we are getting the transfer function, different people uh, from whatever, age and gender and so on, their brain is connected like in similar ways, right? Like the anatomy of a person does not change from person to person that much, right? There is variation. Similarly, I think the functional architecture also doesn't change from person to person. And the way we collect this data, the input in some way is like a random input, right? So if the input is a random input, because there is no input, so the input would be a random input, the output that we are getting is like the transfer function. And so when we analyze these data structure, so it is heavily weighted on the underlying functional architecture. And that is what we are observing. Is that any other? If anyone else has any other guesses, please let me know, because um, I wish I knew exactly what is happening. But yeah.
Could it be because we see brain as a predictive machine? So at points of time, our brain is ready to do the function that it has been doing and will be doing. So all the connections in the brain are making you ready for.
Uh, Right, right. I think that's a very good point. But my point is that yes, the brain is a very good predictive machine, but it's also like the predictions are very reasonable, right? It knows how to only handle um, simple predictions, and therefore, when we are um, collecting the data, um, it would be hard to switch it off. Maybe another way to say is that it would be hard to switch off like parts of the network. So I think like most of the functional network have to be active all the time, right? And that's why even when we get stimulus for very short duration, like let's say related to motion or related, I mean, related to motor activity or visual activity, that is enough to generate these signals and keep them connected, right? Like that, I mean, there is a reason why the brain always needs oxygen, right? We cannot shut it down. And in my opinion, to preserve these functional architecture even more than the structural architecture. Because with consciousness, right, the functional connectivity is altered, but the anatomy is not altered, right? And you can give there is now work using uh, psychoslivin. Like, do you know this drug? Mm, Psychos. I'm mispronounced. Psychocyblin. Yeah. Right. So they completely change your connectivity pattern, right? And so they are like disrupting these functional integration through chemicals, right? The other ways by using like TMS and so on, where you can change these inputs. So that is, I feel that is how. Okay, it's 11:42. Um, I want maybe, okay, let me just try something else before I forget. Okay, so these are the students. Um, so maybe what we can do is maybe, Oh, but it is, I repeat, P, P, okay, all right. Okay, let's see. Um, mm, does it sound okay? Who is Adrija? Okay, maybe you could pair up with one of the unregistered students. Or, or, okay, you can join any group you want. Which group you want to join? Which one? Okay, okay, it's going to be tough. Okay, no problem. Okay, I think, all right. Okay, so these are the groups, right? So better this group have a very good presentation because you have 3 students, right? And hmm, all right. Okay, so for your project, as I had mentioned, what we could do is the autism data which is there is like 100 subjects. So before I forget, I should mention that the data is open access data, so you can download, but please do not, you know, submit like don't put it anywhere or misuse the data because just in so now there are, you know, technology that has been developed where they can even make prediction based on your MRI images how this person would look like, right? So the last thing you want is to say, oh, this person is autistic and post their photo on, you know, Instagram or TikTok or something. So um, also, you know, when you're using this data, you should have, do they have the city certificate, like research certificate, like for subject? So for me, for me to do human research, I have to fill up some forms. Let me just show. Right, so this is what it says, that um, like it has some um, like what does it say, that what it so what it does is that you have to register and basically pass a course, and in the course what is covered is that, you know, these are highly private information. Even if it's open data, you should not share it with someone unless they also have, right? And um, these days, even sharing data is becoming less and not allowed. So you can use it, but after the course, please be careful about not do not share with anyone. Okay, that's one thing. The other thing I would mention is that if someone wants to, rather than autism, wants to do on a different data set, let me know. Okay, it should be okay. It should be okay. But just let me know. Also, um, I'm like I'm going to cover many different methods, right? There are like ALFF, FALFF, REHO, functional connectivity, ICA, like just looking at the standard deviation, and there are many different methods, right? And on top of that, there are the clinical measures. So I would be very surprised if let's say two group gave the same or similar presentation, right? The results from each of the group should be different, because it's like you have to define what the question is and then how you approached it. As long as you did, it would be good. Right? Any questions? Yeah. Which my slides? Oh, okay. Okay, no problem. Don't share it though. Okay. Bec- uh, it's no problem, but I have some some of the I have some figures which has not been published. So no, it's okay if you want. It's okay, but if there is some specific paper you want, let me know. In the afternoon, I can share it. Should we just stop now or continue? Right. It, well, I because it's 11:51. It's like, do I have to continue till 12 or okay? Yeah, maybe try. Okay. Yeah. Yes. Yeah. 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 Right. 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 So we think because during some of the the default mode is not too far from there and may be related, and that is how it is connected, we think. Also, before I forget, for your presentation, send me your presentation like Wednesday, right? Uh, like next Wednesday, send me your presentation. I can uh, like then upload all of this here, and then you can present. Okay. Any other question? Yeah. Yeah. Right, 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 right. That, yeah, right. So maybe I should have made it more clear. So when we compute the standard deviation of a resting state signal, we are, when you compute the mean or the standard deviation, you are throwing away your temporal, you're not using your temporal information, right? So we are only using like a first order statistics, right? We don't have the temporal information, but resting state uses, when we do like correlation analysis, it uses the temporal structure between different region to find relationship. When we are doing the standard deviation, we are saying that the resting state is also affected by the underlying hemodynamics. But because this person is not actively performing some task, at least in the regions where there was task activation, region when they performed a task, and the region which was associated, in those regions during resting state there is no additional activity. So whatever activity there is also getting affected by the underlying hemodynamics, and we are simply taking that and normalizing with the task response, right? So again, this is not the best way to do calibration, but this is like you are getting for free. If you did resting state and you had task, you would get this information for free. Right? Yeah,
Just one thing, we have a huge, because we have a huge data set for 1,047.
yeah,
Are there any differences based upon the languages between language translation
right.
different?
Y- Right. So there has been resting state. Again, like these are really good questions, and thank you for asking. So yes, like there is, like if you are left-handed or right-handed, your associated sensory motor cortex is also more dominant, right? By looking at the connectivity pattern, you will be able to tell, if someone is left-handed or right-handed, right? However, these effects are small, right? These are not huge effect. First of all, the connectivity pattern, during resting state is small, and then between left and right, there is a little difference. So we are always struggling with signal to noise ratio. And that is why many of the earlier studies, they would only consider right-handed, right? So like another example is autism, right? In autism, because like for every 5 boys, there is one girl, right? So the ratio is like skewed against boys, right? That they would get autism. So many centers, what they did was they would not collect females with autism, right? So suddenly there is very little information about what is, how does connectivity pattern look in female with autism? Is there a difference between male and female? So all that information is not clearly known, right? Because like it would be like one has to actively recruit and so on, which is being done now. Similarly is handedness. So there are many handedness study, but they would say, okay, I'm going to collect 20 left-handed versus 20 right-handed and see the difference in the sensory motor activity or cerebellum and so on, right? So many, many of there are differences, but many of these differences is a little hard to find because there's not that many left-handed subjects because people would just have traditionally not included. But now there is a big push, especially in clinical population, for, like developmentally, to include those subjects. So they are being used, right? So like for now for NIH studies, right? Um, like for example, most of the rats were like male rats, which was um, studied before. So now they have to, like any grant you get, you have to study male and female unless you have a very good reason that you have to justify. And even then, it's not that easy, right? So as these things become like more inclusive, then maybe one can find these differences. I think with the UK Biobank, because they have so many subjects. So now it is called community sampling, right? So they will say, we will take subjects from this place and they will take all kinds of subject, right? And they will get clinical and behavioral measures. And when and these information would be available for someone who has access to the data, and then they can look at these differences. Okay. It's like 10, so it would be one, like, so like 11, right? So 10 minutes each is 110. So like 2 hours, that would be okay. Okay. Hmm? It's up to you. Like, do something related to resting state data, right? Don't do a VBM analysis and uh, present, right? It should be something with resting state. All right. All of you have noted down your like group name, right? Okay. Okay. And perhaps it would be good to like run through FSL and other things so that when we come, we could be a little faster today. Like I have to finish by 12. Oh, sorry, 4.